Thank you. There are 300 million people estimated to be living with hepatitis B infection worldwide. This is a huge global public health threat, and it's been neglected by funding, by advocacy, by education, by research, and by clinical practice. There is a pressing need for us to focus on this challenge. So welcome to the crash course in hepatitis B infection. As per the introduction, my name's Philippa Matthews. I'm a clinician. I work in infectious diseases and microbiology as a clinical doctor, and I also lead the hepatitis B elimination laboratory here, working with a brilliant team here in London at the Crick, but also with colleagues and partnerships nationally and internationally. So we're going to go back to the basics to start with. What is hepatitis? Well, let's start with the liver. So the liver is a critical organ. You can't live without it. It's sometimes regarded as the processing factory of the body. And the word hepatitis just means liver inflammation. And there are lots of potential causes for this, one of which is a variety of viral infections. And rather unimaginatively, some of these have been labeled A, B, C, D, and E. We're here today to talk about hepatitis B. Hepatitis B virus, sometimes abbreviated to Hep B or HBV. So having lived through a viral pandemic, we've been exposed to all sorts of images of viruses with and without goggly eyes. Some definitions of a virus. This is one that I like, a piece of bad news wrapped up in protein. A more technical definition is this one, a submicroscopic particle. So this means that viruses are too small to see with a conventional light microscope, typically. And they occupy this interesting boundary between living and non-living. And that's because they can replicate and they have genetic code, so we might think of them as being a living organism. But also, they can't survive independently to replicate. They need to take over the machinery of a host cell in order to do that. We can see images of viruses using a more powerful microscope called an electron microscope, and this is a picture taken using such a microscope of the blood of someone who's living with hepatitis B infection. And you can see that little round particle indicated with the yellow dashed lines, which is the hepatitis B virus particle. But also in that image, you can see all sorts of other funny shapes. And that's because when the hepatitis B virus is replicating, it also generates a lot of extra protein, which gets chucked out into the bloodstream. And it's thought that this is a way that the virus has evolved to sort of distract the immune system, so the virus itself can sneak under the radar. So there's huge quantities of this protein that's called surface antigen. If we take another view of the virus as a sort of cartoon image, you can see this surface antigen around the outside. There's a core protein which wraps up the inside parts of the virus. And there are enzymes. These are the biologically active proteins that allow the virus to replicate. And inside there's a circular molecule of DNA. That's the virus's genetic code. And these enzymes are responsible for driving the generation of new DNA and new viral particles so that the virus can replicate itself. You might remember or know that DNA is made up of these pairs of bases. The bases are called A, T, C, and G. We'll come back to that later. And if we look at the size of the hepatitis B genome, it's actually one of the smallest of all viruses, just 3,200 of these base pairs. And just to put that in some context, that's about 1 million times smaller than the human genome. You might recognize this image. This was drawn in the 1830s by a chap called Charles Darwin. And Darwin constructed that as he was starting to think about the way that all living things are interrelated. And actually, this applies equally to viruses. So small changes happen in the viral genetic code as they replicate. And actually, we've lived through a real-time example of this happening with COVID, right? Because the SARS-CoV-2 virus, that coronavirus, has evolved and changed quite quickly over time, and we might even know which variants we've been infected with. So they're all SARS-CoV-2, but there are lots of different variants and what we call lineages. In the same way, we can construct a tree of hepatitis B viruses. This is all hepatitis B, but the viruses are slightly different due to changes in the genetic code. And this might be relevant because depending on which of those clusters of virus you're affected by, it can actually affect the outcomes of the infection, and it can potentially affect the treatment responses as well. And it's also interesting that these variants are distributed geographically. So depending where in the world your hepatitis B is acquired, you might have slightly different variants and different outcomes. So where did hepatitis B come from? Well, it's part of a wider family of viruses called hepatinoviruses, which also infect birds, amphibians, and other mammals. And we think that hepatitis B jumped from those other species into humans right from the earliest human populations. One example of this in history is the Bronze Age. The Bronze Age, fine jewelry, beautiful art, and hepatitis B. And actually, we now know from sequencing ancient remains that probably at least 10% of the populations of Eurasia were living with hepatitis B during the Bronze Age. Of course, these beautiful people had no idea that what was causing their liver disease was hepatitis B. And in fact, the discovery's only been made in the last 50 years. And it was really led by this man, Baruch Blumberg, who described what he called Australia antigen, studying populations in Australia. And what he'd actually detected in their blood was the hepatitis B surface antigen. And that's what we now still use as a screening test to diagnose the infection, to detect if someone's living with it. He also recognized that there was a major relationship between hepatitis B and liver cancer, which I'll talk more about. And importantly, his findings have led us on to a hepatitis B vaccine. And for these discoveries, he was appropriately awarded the Nobel Prize. So let's just think about how this virus is transmitted. Well, I've said that it replicates in the liver, but it spills out into the bloodstream. And so all of the bodily fluids are potentially affected by the virus, and it's typically spread either through sexual contact or through blood. And blood contact can actually be very minor, so it can be transmitted through a very small amount of blood. But the commonest route now worldwide is from mother to child at the time of birth. And this is really important because infants infected at the time of birth have more than a 90% chance of developing on into a lifelong, that is chronic hepatitis B infection with downstream consequences. So we've been doing some work in our clinic to see if we can detect hepatitis B in small volumes of blood and use these very tiny blood vials to help us with monitoring patients. So this is a picture of tiny blood bottles collectively containing about 1 ml of blood. So I'm now going to ask for some audience participation. So if you've got a mobile phone with you, you can scan the QR code or you can log in with the details on the screen. If you haven't, then I just ask you to look at the question and make a decision about what you think. So the question is, in that 1 ml of blood, how many hepatitis B virions do you think you could find? So we've got several options here, with the lowest being 10,000 and the highest being 1,000 million. So I'm seeing a few people have managed to vote, so I'm going to advance. Okay, we've got a mixture, and it's changing over time. Okay, well done. Those of you who picked the green answer, which is actually the highest number, are correct. So well done to those of you who chose that number. In truth, the number can go much higher, so you can have one with 14 zeros on the end. That's more than the population of the world number of viruses in 1 ml of blood, which is pretty extraordinary. The other interesting thing is that in people living with hepatitis B, although we can have those extremely high viral loads, you can also have a viral load, a level of the virus that's almost too low to detect or measure. So, in fact, those of you who answered with other answers are right in a way, because you can have, there's a whole spectrum. So what happens after you're infected? Well, there's a variety of outcomes, which is really interesting, and these are interesting questions for research. It's actually entirely possible for the immune system to deal quite well with hepatitis B and for it to get mopped up. And for most people in whom that happens, that's the end of it. However, as we've said, and particularly for infants infected early in life, the infection can persist long term. And the reason for this is that the virus archives itself. It makes a very stable copy in the nucleus of infected liver cells where it hides from the immune system and from treatment. And the reason that cure on this slide has got a question mark at the end is because we're still not there. We still don't have strategies to treat that cellular reservoir of hepatitis B. So let's talk about chronic, that's long-term infection. There are different outcomes here as well, also all beginning with C as it happens. You can control the virus. That means it's possible to live with the virus lifelong and it never causes any ongoing complications. But it's also possible, and this is the problem, that people progress down a pathway of liver disease with liver inflammation, liver fibrosis, that's scarring, leading to cirrhosis, that's serious scarring that affects the function of the liver, and then potentially leading to liver cancer. And as you can see on the image, actually, cancers can also interestingly arise in the absence of that inflammatory and fibrotic process. So liver cancers pop up in what otherwise looks like healthy liver tissue. And this is also challenging because for many individuals, this is a silent infection, so there are no symptoms until you reach those severe stages of liver disease. And liver cancer, at least in large part due to hepatitis infection, is the fastest rising and now fourth commonest cause of cancer worldwide. So why do we have this vast spectrum of different outcomes? Well, the truth is we don't completely know yet, but what we understand is that there's a balance between the virus, so particularly the genetic code of the virus, features of the host, that's the person infected, some of which are stable and some of which might change over time, and features of the environment. But the point really is that this virus infects 300 million people worldwide, all of whom are individuals. And what I'm going to do now is pass over to my colleague, who is Joy Coe. Joy and I work together at Mortimer Market Centre, and she's going to tell us a little bit about her own journey.
My name is Joy. I'm the peer support worker for hepatitis B working in Mortimer Market Centre of NHS. I was diagnosed 20 years ago when I was in university after I donated blood to Red Cross, and then I received a letter that I have a hepatitis B. At that time, I have no idea about hepatitis B. What is that? And then I just Google it and then found it, oh my God, it's so scary. You're going to die of liver cancer. I went for my first clinic by myself without any support. And then at that time, I also have a relationship with my boyfriend. And then after I told him, he just rejected me from the relationship, saying that he loves me a lot, but I'm not healthy. And then we will not have any future. This experience is really, really traumatized. Yeah, and then I really feel like I was invaluable person anymore. So I hide my diagnosis from others. I didn't tell any others I have hepatitis B. And then also I disconnect myself from the clinic care. I feel a lot of shame of having hepatitis B until I met my husband who understands hepatitis B really well. He knows that hepatitis B, the most important thing is you need to have a regular monitoring. So he encouraged me, give me a lot of support, and then it is him to bring me back to the clinical care again. And then later we have children, yeah, and then we have a healthy family now. The people living with hepatitis B commonly face 3 challenges: the knowledge gap, and then also the stigma, and also finally it's about the fear of disease progression. As the patients, even though we diagnose, it doesn't mean that we understand what hepatitis B is. And then also the community, also lack of understanding.
So that's a pretty compelling story, isn't it? That leads me on into part 2, which is what we're going to do about this. So we do have good medications for hepatitis B, and traditionally those have just been prescribed for the people that we think are at the highest risk. So not everyone would be eligible for treatment. The treatments that we use are called tenofovir or entecavir, and they block that replication process of the virus. And the good thing is these are safe, they're generally free of side effects, they're easy to take, and they're on the World Health Organization list of essential medications, which means they should be available everywhere. But they're not curative because of that cellular reservoir of hepatitis B, which these drugs don't get rid of. Another great thing about these medications is they can be taken safely during pregnancy and breastfeeding. And that means that as well as being used for treatment, we can actually use them to prevent new infections, reducing the risk of that transmission to babies. We've lived through a really interesting period where there's been this big debate around how many people should be eligible for hepatitis B treatment. And on the one hand, we've got all of these benefits that we've talked about in reducing liver disease, also potentially reducing the mental health components of hepatitis B infection, of anxiety and stigma, and tackling transmission. But on the other hand, if we're really going to treat 300 million people, that's quite a challenge. There are logistics and costs involved. We might be treating some people unnecessarily who wouldn't go on to get complications, and not everyone either needs or wants to take long-term treatment. And so we've also got to consider the balance for individuals and also across populations. What's the balance of risks and benefits? And the WHO has been considering this and has now produced new guidelines earlier in 2024, which are a real turning point, because we've gone from this environment where only a minority of people are offered treatment and the eligibility criteria are quite stringent, to a much more relaxed environment where many more people will be offered treatment, and the needle can even swing so far as to say we could treat everybody living with hepatitis B. In England, we've had this really amazing programme rolled out over the last 2 years through NHS England, where emergency departments since April 2022 have offered opt-out testing for blood-borne viruses. So that's hepatitis B along with hepatitis C and HIV. And as a result of this programme, nearly 2,000 new diagnoses of hepatitis B have been made. And we know that many of these are in people who would have otherwise not had access to a diagnostic test. But at the moment, we're lacking really robust pathways to understand how we can feed those individuals into a meaningful care system that's sustainable and can offer long-term treatment. So there's still work to be done. I've mentioned the vaccine, and now all infants born worldwide should be having at least 3 doses of hepatitis B vaccine, and in high-risk settings, they should also get a birth dose hepatitis B vaccine. And the vaccine is made up of this surface antigen, so that envelope protein of the virus, given alone, so there's no DNA, it's not infectious, but it makes a really robust immune response. The vaccine, like drugs, is actually really safe, effective, cheap, very minimum side effects, and it should also be available, as well as to babies and infants, it should also be available to other groups at high risk. The caveats here are that we need at least 3 or 4 doses, and there have been big gaps in distribution, so there's a real problem in equity and reaching the populations who most need it. Nevertheless, since the 1980s, more than 1 billion doses of hepatitis B vaccine have been distributed worldwide. And I was pleased recently to be in South Africa in a pharmacy up in the north of KwaZulu-Natal, and finding hepatitis B vaccines in the fridge is a good moment worth taking a photograph of. You may think there are more exciting things to find in a fridge, but actually Hep B vaccine's up there for me. So the vaccines reduce individual infection, they reduce liver disease, they reduce liver cancer, they reduce transmission. All of this saves money long-term for healthcare systems. And another great example of this is the human papillomavirus, HPV vaccine, tackling a different virus, but with a similar consequence on cancer, because HPV is a main driving cause of cervical cancer. And what we've seen in young women particularly is this drop in cancers caused by HPV as a result of vaccine programs. For hepatitis B, we're failing to deliver this adequately at the moment, with only 45% of babies worldwide receiving that crucial birth dose vaccine. So that leads me on to tackle some of the problems and challenges that we're facing. This is a particularly major problem in Africa, and the hippos highlight that. We'll come back to them in a minute. So I've said that this is a major global public health threat, and in fact it's up there with HIV, malaria and TB, but it's been really neglected. Although we have this population, as I've said, of 300 million people living with the infection, we've not tackled it adequately. 12% of those are children, which is really unconscionable in an era of a safe and effective vaccine. While I'm giving this talk, we can expect that 100 people worldwide will have died from their hepatitis B infection, and that's what leads us to over 1 million deaths each and every year from what is, in fact, a preventable and treatable infection. And the reason for this is partly that we're dealing with not the tip of the iceberg, but the tip of the hippo. We're failing to diagnose people, so most of those diagnosed are not aware of their status. We're failing to link people into care, so many of those living with hepatitis B aren't connected to the right care pathways, and we're failing to offer and deliver effective antiviral treatment. So in the context of all of this, there are some very ambitious global goals that have been agreed for the elimination of hepatitis B as a public health threat by the year 2030. It's not far off. We're aiming for a 90% reduction in new infections, we're aiming for 65% reduction in deaths, and we're aiming to reduce hepatitis B infections in children. Some of those targets in some populations will be achieved, indeed have been achieved already, but in many places we're very far off achieving these goals. One of the reasons for this is lack of research in the right place. So this is a map showing the distribution of hepatitis B infection worldwide, with the red and orange countries the most highly affected. And this data is put together by Marianne Delphin in my group at the Crick, looking at where hepatitis B research is done using the same colour scale. So if we look at Africa, the highest burden countries in red and orange are in fact blue and green on the clinical trials map, showing that there's no data, no clinical trials representing these countries at high burden. We've also thought about this at a more individual level, and Joy's personal story gave us many insights into that. These are some voices and stories that we've collected from the east coast of Kenya, reporting people's journeys living with hepatitis B. Most of the patients come to hospital very late, so late that most of them die. Those infected are isolated. Some of them believe that they've been bewitched. Having a hepatitis B diagnosis will bring shame to one's family. So these are all stories about the individual impact, but also a wider societal and lifelong impact of this infection. These stories aren't just true in Africa, they're true on our doorstep here. So this is a picture of the Crick Institute where we're gathered now, and University College London, which is 15 minutes up the road, where I'll be walking later to do my hepatitis clinic. And on that walk, I am reminded every time of the very profound inequities that still exist in our society. Many of these people suffering from a variety of marginalisation, vulnerability, economic poverty, also live with chronic health conditions, including hepatitis B virus. And Emily Martin put together this paper about the forgotten people. That's a quote from an individual living with hepatitis B infection describing themselves. And so we're working in partnership with the Inclusion Health Programme here in London to try and recognise and redress these challenges. Let's think about cost. We know that the global benchmark price for tenofovir should be $2.40 a month. That's sort of half a cup of coffee in London, if you're lucky. But these costs vary widely. These are data put together by the Clinton Healthcare Access Initiative showing the monthly cost of treatment. And what you can very easily see with that is that these costs can become catastrophic for people. There's an official definition of catastrophic costs, which is an economic cost exceeding 20% of annual household income. And when you look at people living on less than $1 a day, many of these are in Central Africa, then catastrophic costs are incurred very easily by living with hepatitis B infection. That's just the cost of the drug. It's not factoring in all the other costs which you might be paying for blood tests, hospital visits, time missed from work, etc. So that leads me to the challenge of what we're actually doing about it, which is the final part of the talk. So these are some pictures of my group here in action at the Crick, some laboratory studies and lots of other action going on. We're working with these wonderful partners. Louise Downs is a clinical fellow funded by the Wellcome Trust who set up a hepatitis B research program in Kilifi in Kenya. We're working with a big community program in Uganda studying liver disease. We've got a fantastic partnership with Stellenbosch University. That's my friend and colleague Tongai Maponga, with whom I've worked for many years now. And more recently, and supported by funding from the Crick, we've developed a partnership with the Africa Health Research Institute. I'll show you a bit more about that now. So Motswedi and Gloria, who are shown on the slide here, are core members of my research team, but based permanently in Durban in KwaZulu-Natal, which is the province shaded red on this map. And the population here live with lots of health challenges, as you can see from those data, in terms of HIV infection, low rates of education, high rates of unemployment. And our data at the moment suggests that about 11% of this population is living with hepatitis B. A third of the population has been infected at some point, so the rates of exposure to this virus are still very high. And less than 10% of the population has got evidence of having been vaccinated. So there's a big gap. We've got a big public health problem. We're still failing to get the vaccines out there. And we can think of this in terms of a care cascade, which is this sort of diagrammatic model of who's diagnosed and then who's been linked to care, offered treatment, and is having successful treatment. And there's a big drop-off at each step of the pathway. And that's due to interplay between a whole range of different factors that are listed on the slide here. So what we're trying to do is set up programs that tackle all of these different components, all of these different barriers. So we're using this community screening that I've already described. We're engaging with the public, and we're then inviting people living with hepatitis B to come for clinical review. We can then collect samples for laboratory studies, and we can put all this together to try and develop better packages of clinical care. So there are 4 things on this list, and I'll say something brief about each of them. So first is understanding the social context. So we've gathered people together as part of our research teams, and then we've invited members of the community to come and tell us about their experiences and the barriers that they experience to care. They've told us how there's poor understanding and knowledge. They've told us about challenges in facing stigma and discrimination, including from healthcare workers. And they've told us quite correctly that liver disease has very poor outcomes. And some of this has been written up and published as a research paper. We've also engaged with school students, both in South Africa and in the UK, who've told us about their experiences, their connections to healthcare, and their understandings of vaccines. And I've just got a tiny clip here, which is a student in South Africa telling us about the use of traditional medicines in her community.
Is this traditional when you are sick or when you have a fever?
So you can hear that she's talking about a traditional herb that's taken when you're sick or when you have a fever. And actually lots of the drawings produced by the students and the testimonies they produce tell us about their use of these kind of medications. So we're also trying to build better bridges to traditional practitioners and healers so that we can use that as a way of engaging with the community. Engagement with the community is also facilitated by better communication, and we've been making these visual materials to try and provide accurate and easy to access information in the right local languages. We're also thinking about how we can deliver better testing and screening into communities. This machine in the pictures is called a FibroScan. It's based on an ultrasound machine, and it allows you to do a very quick and easy scan of the liver, which gives you a measure of stiffness, liver stiffness or inflammation. But the problem is, again, healthcare barriers of the people who we've trained and engaged with this equipment. Most of them say it's the cost of the equipment that's the main barrier to further delivery. What about delivery in our London population team I've mentioned? Well, my colleague Indrajit Ghosh has designed and had built this prototype clinic on a bike. This is the world's only at the moment. Here he is on the streets of London, taking this out to reach communities and individuals for whom our healthcare services are otherwise inaccessible. Let's hear Joy talking about her role in bringing clinical care to populations in London.
My day-to-day work as a peer support worker for hepatitis B is actually about the human touch. I think my presence, the most important thing is we talk about it. Yeah, because I believe most of the patients, they just living in the dark corner, never talk about it with anyone. So my role is that we are equal. We have same diagnosis. We can talk about it very naturally with very supportive environment because I understand the whole journey. I believe it is very important to let the research team hear our voice. I think we, as the patient, we would really like to be involved in the research, but I think the problem is not every patient have equal access to the same information. So I think definitely for the research team, they need to enter our world and to engage with us.
So that's a clear message for us, right? Which is that as clinicians and researchers, we need to do much more to engage with the patient voice and the patient journey. And that leads me into a slightly different approach to engaging with patient data, which is through this program called the Health Informatics Collaborative, which has allowed us to collect and share data from a number of hospitals around the UK. The data are collected, cleaned, anonymised, held in a safe environment where they can be reused to address research questions. And we can then use that to lead us into improvements in care provision. So we've now got data from over 23,000 adults living with viral hepatitis, in particular hepatitis B, across the UK. And since we're living through this big change in the guidelines and in clinical prescribing, we can use these data to our advantage to look at, for instance, how many people are currently being treated, how many will be eligible for treatment as the guidelines change. And you can see what happens. We're currently treating about 25% of our population. As changes in guidelines are rolled out, we might be treating 50%. We could even move towards a treat-all kind of model. And this is very helpful in planning the way that our healthcare is delivered. And of course, there are lots of other questions we can ask of big data. What are the long-term outcomes? What will happen as new drugs become available? And there's also a consortium in which we're involved in Africa, collecting similar data and feeding that again into a whole pool so that we can start to get the big picture. So individual factors, let's just think about the environment in which we exist. This is an original Grayson's Cafe donut from the Crick. I had to buy it to support the development of this talk. But we all essentially now live in this environment where we're exposed to way too much of this stuff, right? And it's not good for us. And we know that fatty liver disease, metabolic liver disease is a big contributor to a population health explosion of liver disease. We can measure fatty liver or steatosis, again using a simple ultrasound-based technique. And we can also measure this range of other factors which put stress on our livers. And we've been doing that in our local clinic here in London. And actually, we've converted the Grayson's donut into a donut plot, which shows you that 22% of our patients here locally are living with moderate or severe fatty liver disease on top of hepatitis B. And around 2/3 of them have at least one of those other liver stressors going on in the background. What we don't currently know or understand properly is what is the interaction of these? How do these healthcare challenges influence each other? And what should we be doing about it? So we've done some description, but we're now setting out to understand and actually tackle, find ways that we can intervene to improve liver health. And here at the Crick, we're very lucky to be able to work in partnership with big technology platforms that offer us access to new testing strategies. The metabolomics program led here by James McRae allows us to take tiny samples down to 5 or 10 microliters, that's a fraction of a mil, and measure a whole detailed profile of blood lipids. And we've got this work underway. We're also working in partnership with a technical training fellow from the Crick African Network so that we can develop capacity, help with training, and answer research questions together. And my final slide on the science is to think about going back to that tree of hepatitis B viruses. We can actually now read hepatitis B viral sequences. This is work done by Sheila Lumley alongside lots of colleagues and collaborators. And we're using a tool called nanopore, which unzips that DNA strand, feeds it through a microscopic pore in a membrane, and reads the electrical current. And remarkably, that gives you the genetic sequence of anything, in this case hepatitis B, but it's a great way to generate sequences of any living organism. And we can then identify from blood samples from people living with hepatitis B how the sequence of the virus might affect viral load, might affect liver disease outcomes, might affect responses to vaccines now or in future, might affect the way the virus is transmitted, and can have an impact on treatment efficacy. And the treatment efficacy is something that we're particularly focused on. So Khadija here at the Crick has been working really hard to set up laboratory assays where we can try different variants of hepatitis B in culture. We can then explore the effects of different drugs, different doses and different combinations to really understand and be able to measure in a very controlled setup what's the impact of treatment. And lots of these conversations are feeding back into dialogue with the World Health Organization, who are setting up a new global action plan, looking in particular at drug resistance, so thinking about what the outcomes of treatment are and whether there are Achilles heels in the treatments that we're currently using. So I've covered a lot of ground, and it's now your turn again. So mobile phones back out if you've got them. I'm going to give you a hypothetical scenario, if only this were true, that we were given a large pot of funding for hepatitis B, but you're only allowed to choose one category. So I'd like you to cast your votes. It's nice watching this evolve in real time. Don't get influenced by others, cast your own vote. Okay, well, it looks like vaccines are winning. I don't disagree with that. Of course, what we've actually got to do is all of this. No one of these strategies is enough on its own, and we've got to invest in all of this together. And bringing together these multidisciplinary programs allows us to do that. So, to conclude, we have got so many tools at our disposal. We've got this wonderful shift in the landscape, especially in terms of diagnosis and treatment, and we've got some elimination targets that are reachable. But to look at the glass half empty, we are still living in the context of a high global burden, a lot of unanswered questions, and we're undoubtedly off track towards global elimination targets. This headline from The Guardian puts it in the best way I can think of. Tackling this is a thundering moral imperative. Let's just hear from Joy to close.
Definitely believe the research outcome, the result will be much better with lots of patients involved.
So there we go. The challenge is set. So I'm going to conclude by thanking you all very much for coming, by thanking this wonderful team of individuals who champion, support, lead, inspire this work every day, and so many of our collaborating centres. Thank you all very much for your attention.